This will be a video about B1 6.1 and B2 6.3. So it will be about gene basics and genes and alleles. You will find it in your book on page 72, 73 and page 158 and 159. After watching this video, you will be able to describe how genes control characteristics, describe how genes are located on chromosomes, describe how the nucleus contains those chromosomes, and you will be able to define the terms chromosome, DNA, gene, locus, allele, and gametes. You will be able to explain why different alleles produce different characteristics and explain why sexual reproduction produces variation in the offspring. All our DNA is stored in the nucleus of our cell. DNA is a very long molecule. If you would take all the DNA from one human cell, it would have the total length of 2 meters. So in order to make all the DNA fit in the nucleus, the DNA is coiled, it's supercoiled, and it's also wrapped around proteins called histones. On one chromosome you will find many genes. And there's, for example, a gene for the protein that we find in earwax. But there's also a gene for eye color. So a gene is a part of a chromosome that codes for a characteristic located on one chromosome. And the place where they're located, we call that the locus. So the place of a gene on a chromosome is called locus. The plural of locus is loci. For certain genes we have more than one version. For example, there is a gene version that codes for blue eyes, but there's also one that codes for brown eyes. Those different versions, we call them alleles, and an allele is a version of a gene. So we have an allele for blue eyes and we have an allele for brown eyes. You might have heard about the nature versus nurture debate. It's always the question, are we the way we are because of our genes or because of our lifestyle? Our genes, they determine a lot of things about us. That I have blue eyes is because of my genes and I can't do anything about that. My eye color is basically decided by my genotype. Genotype, another word, is genome. That I have a belly is because I eat too much. We call that your phenotype. And the phenotype is the result of your genotype and the environment. For some characteristics, the lifestyle or the environment is very important. So the genotype plays a role, but the environment is also very important. Now, in these cases, you will have a continuous variation. Height is an example of that, or hair color. But there are other characteristics that mainly are influenced by the genotype and less by the environment. In these kind of situations, the variation is categoric. You can separate them in different groups. Either you have 3 earlobes or they are attached, or you have blue eyes or brown eyes. So in this case, the genotype is more important than the environment. Genes code for proteins. What are proteins? Proteins are very long molecules and they are made up out of amino acids. So if you put many amino acids, if you combine them as you combine beads in a necklace, you will make a protein, and that's all the DNA codes for. So our DNA is transcribed into RNA, and then there are some big enzymes. We call them ribosomes, and they can read the DNA. And what they do, they just combine the necessary amino acids to form a protein. This is a process that you don't really have to understand yet. The only thing you have to know now is that the DNA codes for proteins. Proteins are very important in our body. All those big structures you see here in the cell membrane, they are proteins. So the proteins, they decide what will enter the cell and what will leave it. But also enzymes, they are proteins. So proteins are very important, and that's why you can make an entire human body when you know which proteins you have to make. Let's have a look at DNA profiling, or DNA fingerprinting as it is also called, many times used in series as CSI, or you can find out the kinship between individuals, so who's the father, or these kind of things. Now, how does it work? If you take a DNA sample, or a blood sample, or a cheek swab, you can extract the DNA from that, and you can add certain enzymes that cut at specific places in the DNA. And because of that, you will have a mixture of DNA fragments with different sizes. If you take that sample with those different fragments and you put it on a gel, you can do an electrophoresis, and you will separate them based on their size. Nowadays they use many times gas chromatography for that. You can also use the gel electrophoresis, which is a very simple technique to use. Let's have a look at this situation. There's a mother and she doesn't know who the father is. For some reason there are 3 candidates, and we have to find out who is the biological father. The DNA of the child is coming from the mother, and the father half is coming from the mother, the other half is coming from the father. That means that those lines, those bands that we can see, are either coming from the mother, and if they are not coming from the mother, they have to be coming from a father. So let's have a look first at all the bands that can be explained by the profile of the mother. Now you see there are 3 lines that overlap. And there are 3 that are not. So those 3 have to come from the father. Let's have a look if we can find all 3 in the profile of those candidates. Now in the first one you can see it only explains 1, but the other 2 are not coming from him, so that means he can't be the father. And this one you can see, number 2, there are 2 bands that can be explained like this, but still 1 not, so he is not the father. And number 3, yes, that is bingo. You can see all 3 lines are coming from him, so that means that number 3 is the real father.